So this next video is going to be about membrane proteins and what are involved with these. So some of these terms you might have heard of before, but if you haven't, we'll kind of go over them with a quick example. Solute is a substance that dissolves in a solvent. A solvent is a substance that dissolves in a solute, and a solution is basically made up of the solvent and solute mixed together. I know that sounds really difficult, but you do this all the time. So imagine you're going to go and make a cup of coffee. So you have the coffee, which is the solvent, and let's say you're going to put either sugar or cream in it. So those two could be the solute. So let's just focus on the cream. You put some cream in it and you mix it. That becomes the solution. So the solution is the coffee with the creamer in it. It could have easily been the coffee with the sugar in it, and the sugar would be the solute. So basically it's just saying, what is the liquid, what are you mixing in it, and when you do mix it in it, it's called a solution. So blood is a solution, and what's mixed in it? The solvent would mostly be water, and the solute could be, there's a ton, gases, sugars, all these different things that kind of go in to make blood what it is. Of course, red blood cells. Concentration is a measure of the amount of solute dissolved in the solvent. So example, blood has a salt concentration of about 9 grams per liter. So if the salt levels are too high, you have a major problem. If the salt levels are too low, you have a major problem. And your body has stockpiles of the salt, so it just does this thing called homeostasis, where it either can get to it or get you to crave some foods with salt in it so it can get that level back on track. And then the last one is a concentration gradient. The difference between a concentration of a particular substance between two areas. So for instance, if the inside of the cell had more water than the outside of the cell, that will be a concentration gradient. And then what will eventually happen is that water will flow from one direction to the other direction. So that's the concentration gradient they're sort of talking about. So even though these terms are all bolded and red and they look kind of scary, they're actually pretty easy. What we'll find with a cell is that there are areas that have higher concentration of one thing on one side, lower on the other side. And we have to adjust for that. So for instance, one of the ways we can adjust for that is this thing called simple diffusion. And look, there's our friend, the lipid bilayer again. Remember, areas that love water, areas that hate water, and areas that love water again. So let's say this is the outside of the cell, this is the inside of the cell. The movement of these particles from an area of high concentration to an area of low concentration is going to basically make things go from high to low, and eventually they want to get to equilibrium. So they're going to be, right now you can see there's a high amount of these molecules on one side and low amount on the other side. You can see that they're going to flow this way, flow inside the cell, until eventually they're equal outside and inside, and then they'll sort of stop. And that's called simple diffusion. Particles move down with the concentration gradient. It happens across a plasma membrane. It doesn't really need a plasma membrane because diffusion happens anywhere. We could use the example of having perfume. If someone in the room has a lot of cologne or perfume, it'll start out in high concentration in one area, but eventually it'll diffuse throughout the whole room if we give enough time and get to equilibrium. So it'll be hard to sort of pinpoint who has it after a couple hours, but if you initially came in and sat in the room, you'd be quickly able to tell who had the most perfume because it'll be in high concentration, but over hours it'll be in equilibrium all over the place. So when I think about these guys, I think about simple diffusion, I think about gases, things that don't have a charge on it, so nonpolar items can move this way as well. And those are the things that do that. Facilitated diffusion. Number one, the first thing you could see is that there's our lipid bilayer again, but now we have these proteins. So these are going to move larger things. So these proteins allow the solute to move across a membrane like a door. So the solution still moves down with the concentration gradient from high to low, But basically now we can get around some things. Like for instance, we can let charged particles in. We can let large things like amino acids and nucleotides in. So now all those rules that happened before, it had to be nonpolar. Well, we could still move things this way because these guys could be polar, and it's just going to let this method come in. It could just be that these are really large, so by themselves they can't come through the lipid bilayer, but through this door they definitely can. Osmosis is a type of diffusion, but the only thing, the only thing that can go from high to low is going to be water. So what you see is these little dots, they represent water molecules. And you can see that here we have a high concentration of, oh, actually, no, I'm sorry. This is sugar. So there's a high amount of sugar on one side and a low amount of sugar on the other side. So what's going to happen is, where's there more water? Well, it kind of says it right here. But because there's more sugar on this side, it also means there's less water on this side. Here, there's less sugar, so there's more water. So what's going to happen is water wants to get to equilibrium. So water is going to go from an area of, this area has less water, this area has more water. So water is going to now pass to the other side. So what's going to happen is, just like you're seeing in the specialized beaker, can you see how the water level dropped on this side, but the water level is higher on this side? It's because water wants to rush to this side till they're equal on both sides. So let's do an example. Let's say there's 10% sugar on this side and 1% sugar on this side. How much water is on this side? If there's 10% sugar, there's going to be 90% water. And if there's 1% sugar on this side, there's 99% water. So 90% water, 99% water. That's all we need to do. We need to just cut down to the chase. So I can imagine a question that says, number one, what side has more water? So in your brains, you've now said 90% water on this side, 99 on the other side. Now it's going to say, what's the direction of water going to be, or where's the osmosis going to go, what direction? So you can see that 99 on this side, 90 on this side, they're going to go until eventually they're in equilibrium. So on this side, the water level, and this side, the water amount, concentration is going to be the same. You can see the water level is changing, but the concentration of water will eventually be at equilibrium. And that's what the body wants to do. So this is kind of representing a cell, where a cell has an inside and an outside, and if there's not enough water on the inside of the cell, the water will move in and get to equilibrium. Cells will basically have three states that they're going to be in. Isotonic, that means equal amounts of water inside and outside the cell. Hypertonic, hypertonic is basically extracellular concentration that's greater on one side or the other, and we'll talk about that in a second. What will end up happening is the outside has more water, so the inside will start to let the water out, and the inside will sort of shrink up, kind of like a grape. Like a raisin. Where hypotonic, more water is going to rush in, so what will happen is it'll start to swell, and eventually it will pop and burst. If we look at these pictures, isotonic, these are red blood cell. This is what a red blood cell should look like, with that little dent in it. This means equal amount of water inside and outside. You can see the direction of water is about equal. On this side, it looks like water is only rushing out. So now the cell looks like this. It sort of looks deflated, kind of like when I think about it like a raisin. So it doesn't have that ability anymore, and it's not going to work as well. So it'll actually stop working eventually. So that becomes a problem. Here, hypotonic, more water is rushing in, and because more water is rushing in, remember how it's supposed to have this little dent in it? Now it's completely round. And because it's completely round, it's not going to move around too much, and eventually it's just going to pop and explode, and it'll be useless. So these are the states of what's going on. Here, water is equal on both sides. Here, there's more water on the outside than less on the inside, so the water is rushing out. And here, the water is higher concentration outside than inside, so it's rushing in. When we make drugs, when we make IVs, when we hydrate people, or treat them, we have to keep this in mind, because in the past, we constantly did these sort of things, where you'd have a patient come in, you wouldn't keep cognizant of the blood, the salt solution in blood, and their cells would explode, and of course they would die. Active transport, this one's a little different. This one's going to require protein or energy to work. It's going to use solute to move against the concentration gradient. So now what's going to happen is ATP is like currency. It's going to allow the cell to do some work. And these molecules will bind into the cell, and with this energy be able to be pumped to the other side. When I think about active transport, what I'm thinking about is things going the opposite direction that they normally want to go. We know most things, when we talk about osmosis and diffusion and facultative diffusion, they all went from high to low till they got to equilibrium. Where active transport is going to move things from low to high and never, never, never wants to get to equilibrium. So it's going to push things the opposite direction. So you could think of things like waste. It's going to push those things out. It's going to move some specific ions that we'll talk about later when we talk about muscle movement and things, one direction or the other, against the gradient. And that requires energy to do that.